Hello everyone, today we are from Group 7 are going to present about the polyclonal and monoclonal antibody. So let's move to the introduction part. Do you know what is antibody and how does it look alike? Antibody actually is in Y-shaped form and known as globular protein or immunoglobulin that synthesized by plasma cell in response to present of foreign antigen. So how this antibody work? This antibody will bind to specific antigen and then it will create antigen-antibody interaction. So now we move to the polyclonal and monoclonal antibody. What is monoclonal and polyclonal antibody? Okay, polyclonal antibody actually represent a collection of antibodies from different B cells that recognize multiple epitopes on the same antigen. In contrast, monoclonal antibody represent antibody from a single antibody-producing B cell and therefore it only binds with one unique epitope. This monoclonal antibody also known as monospecific antibody.
You have no idea of what we are talking? Don't worry, we have one video for you. Perhaps after watching the video, you will have more understanding about what monoclonal and polyclonal antibodies are. Hi, I'm monoclonal antibody.
Hey, hi, I'm monoclonal antibody as well.
I will be binding to epitope B.
Oh, I will be binding to epitope B as well.
So let's go and find epitope B. Woohoo!
Oh no, we couldn't bind because it is epitope A.
So let's go and find epitope B. Where is epitope B? Oh, epitope B is here. Woohoo!
These are the two monoclonal antibodies. They are identical to each other because they come from the same lineage of B cell. So they will bind to a unique epitope on the red blood cells or the antigens.
Hello, I'm polyclonal antibody.
Hi, I am polyclonal antibody.
I'll be binding to epitope A.
I will bind to epitope B.
So let's find our epitope.
Okay.
So, these are polyclonal antibodies. They are not identical to each other because they come from different lineages of B cell. So they will bind to different epitopes.
Let's now have a look at our monoclonal and polyclonal antibodies production. Since monoclonal and polyclonal antibodies, they differ from each other, even their production process will differ slightly. So first, let's have a look at the production of monoclonal antibodies. Monoclonal antibodies, they bind to a unique epitope on an antigen. So first, let's take our host animal, in this case is our little rat. We inject it with an immunogen. This process is termed as an immunization process. So after the immunization process, an immune reaction will be generated, and this will lead to the activation of a single lineage of B cell from the spleen. This is actually the hallmark of monoclonal antibody production. That is, only a single lineage of B cell will be activated. So we take that single lineage of B cell, we culture it with myeloma cells in a cell culture media, and they will undergo fusion to give rise to hybridoma cell line. The hybridoma will be normally the one producing the monoclonal antibody. But before that, the hybridomas or the antibody producing hybridomas are cloned, and they represent a stable, renewable source of antibodies because these are the ones that will be generating our antibodies. These antibodies generated are identical to each other, and they are known as the monoclonal antibodies. Like I said before, they will bind to a specific epitope on an antigen. Coming to the production of polyclonal antibodies, the first step is same as monoclonal antibody production. We take our little host animal, our rat, we inject it with another type of immunogen, the immunization process. So after the immunization process, again, an immune reaction will be generated and will lead to the activation of B cells. But in polyclonal antibody production, multiple lineages of B cells are produced, or activated actually, and these B cells will excrete polyclonal antibodies into the blood of our little rat. So in order to obtain these polyclonal antibodies, we simply have to take a whole blood sample from our immunized rat. and we just centrifuge it because normally the polyclonal antibodies are found in the plasma or serum. So after the centrifugation, we can just obtain the serum in which there is our polyclonal antibodies. But if we want just a pure sample of the polyclonal antibodies, we can just purify it and remove the other serum proteins. So here we are, we have our antibodies, polyclonal antibodies, which have varying paratopes and have varying affinity for target proteins. So that was all about monoclonal and polyclonal antibodies production.
Now, let us move on to the different characteristics between polyclonal and monoclonal antibodies. Polyclonal antibody can recognize multiple epitopes on an antigen, whereas monoclonal antibody can only recognize one epitope per antigen, as shown in the diagram. Polyclonal antibodies are inexpensive to produce because they require minimal technology, and this consequently lowers the laboratory costs, as compared to monoantibodies, which are expensive to produce due to the use of higher technology and also trainings. Next, polyclonal antibodies are quick to produce. The purified antibodies are ready to be used approximately within 3 months. This is because the production is less complex as compared to monoclonal antibodies, which require a longer duration to produce, which is approximately within 6 months. Polyclonal antibodies offer greater sensitivity for detecting antigens that are present in low quantities in a sample, since multiple antibodies will bind to multiple epitopes on the antigens. For monoclonal antibodies, they have less sensitivity. This is because small changes in the epitope structure often render the monoclonal antibodies unable to detect the target protein. Since polyclonal antibody can recognize multiple epitopes on an antigen, it prunes to batch-to-batch variability when they are being produced, and also cross-reactivity is likely to occur. For monoclonal antibodies, since they can only recognize one epitope per antigen, they have no or low batch-to-batch variability when creating identical batch during hybridomas. The high sensitivity to a single epitope makes them unlikely to undergo cross-reactivity. Polyclonal antibodies are non-specific antibodies, which are mainly made up of IgG subclass, whereas monoclonal antibodies are specific antibodies which consist of only one type of antibody subtype, for example, IgG1, IgG2, and IgG3.
So now we are already at the summary part. As I told you before, the polyclonal antibody are represented by collections of antibody from different B cells that recognize multiple epitopes on the same antigen. Because of this characteristic, they are first, inexpensive to produce, as the production process is easy and widely available. Second, quick to produce because it only takes approximately 3 months, and the production method usually results in high yield. Third, it highly sensitive because the different antibody present are able to recognize different epitopes, so it is good for screening method. However, this polyclonal antibody have its own disadvantages, which are, first, it able to generate large amount of non-specific antibodies. Why this thing happen? This is due to the polyclonal antibody able to recognize different epitopes on the same antigen. Second, it increase the cross-reactivity with similar antigen. And third, it have low specificity, so it tend to have more background. For monoclonal antibody, because it monospecific antibody and able to recognize one type of antigen only, so here they are the advantages. First, high specificity to a single epitope, thus make them extremely efficient for the binding of an antigen. Second, it reduces the probability of cross-reaction since it only consists of single antibody. Third, it have less background from staining of the cell because it is specifically detecting one target epitope. But the monoclonal antibody also have its own disadvantages, which are, first, expensive to produce as it required more time and high technology for production, and second, it less sensitivity because too specific to detect across range of species. So Now we move to the conclusion part. As a conclusions, both of these antibody have its own advantages and disadvantages. But nowadays, we are more prefer to use monoclonal antibody, especially in blood testing, since it is very specific and can reduce the false positive result compared to the polyclonal antibody that only used for research purposes like ELISA. Thank you everyone.